Hi, I'm Munich. Today I'm going to be presenting on the cyanide ion effects on cellular respiration. First of all, let me introduce you what is cyanide. Cyanide ion, or Cn- consists of C and N. In the nature, it can be found in gas form of HCN or hydrogen cyanide or salt form. For example, sodium cyanide, NaCN. It has high toxicity to the human cellular respiration. Cyanide can be found in form of cyanogenic glycosides from plants such as cassava, bitter almonds, cherry pits, and apple seeds. Generally, cyanide can be used for several industrial benefits such as mining, electroplating, and chemical synthesis. However, exposure to cyanide have negative consequences to health. The toxin itself can inhibit cellular respiration. Moreover, the chronic exposure leads to neurological effects such as weakness, paralysis, and motor dysfunction. Thyroid dysfunction. Cyanide can impair iodine uptake, leading to goiter. And cyanide-induced neuropathy. Prolonged exposure can damage nerve issues. Since we know what is cyanide, let me introduce you what is cellular respiration. Cellular respiration is a process in which cells convert nutrients, or glucose in this case, to be ATP. Before we go further, let me explain you what is ATP. ATP, or adenosine triphosphate, is a primary energy carrier in cells, which provide energy for various biological processes. It consists of a nitrogenous base, adenine, a sugar ribose, and 3 phosphate groups. Energy is released when ATP is hydrolyzed to be ADP, adenosine diphosphate, and inorganic phosphate. ATP is mainly produced in the mitochondria through cellular respiration. It powers muscle contractions, active transport, and biosynthesis in cells. Before we return to cellular respiration map, let me introduce what is NADH and FADH2 as well. NADH, nicotinamide adenine dinucleotide, is an electron carrier in cellular respiration as well. It carries high energy electrons to the electron transport chain ETC in the mitochondria to generate ATP by its donating electrons. Each NADH molecule contributes to produce 3 ATP molecules in an oxidative phosphorylation. FADH2, flavin adenine dinucleotide, is another electron carrier involved in a cellular respiration. Like NADH, FADH2 donates electron to the electron transport chain ATC, but at a lower energy level. Each FADH2 molecule contributes to produce 2 ATP molecules in an oxidative phosphorylation. Then we go back to the cellular respiration. So let's focus on cellular respiration now. Cellular respiration has 2 categories, aerobic and anaerobic. Aerobic respiration occurs when oxygen present, while anaerobic respiration occurs when there is no oxygen present, or cell cannot use oxygen. In fact, the anaerobic respiration has 2 categories, which are lactic acid fermentation, which occur in animal cell, and alcohol fermentation, which occur in plants and yeast. In this case, we are going to focus on lactic acid fermentation in human. Under a normal condition, the cell digests foods with the presence of oxygen. Therefore, aerobic respiration converts glucose to be energy, as shown in this equation below. However, this equation is a net equation of 4 consecutive steps: glycolysis, acetyl-CoA formation, Krebs cycle, and electron transport chain, or ETC. First, glycolysis occurs in cytoplasm. It has 2 substeps: energy investment and payoff. To complete energy investment, the cells will include 2 ATPs into glucose to create fructose 1,6-bisphosphate. Subsequently, at the beginning of energy payoff step, fructose 1,6-bisphosphate with its 6 carbons will be broken down into 2 3-carbon compounds, glyceraldehyde 3-phosphates, or G3P, and dihydroxyacetone phosphate. However, dihydroxyacetone phosphate is unstable, therefore it will be changed to G3P. 2 G3P are converted to be 2 pyruvates, while creating 4 ATP and 2 NADH. As a result, the net glycolysis creates 2 NADH and 2 ATP from substrate level phosphorylation. A substrate level phosphorylation means a process of ATP generation where a phosphate group is directly transferred from a high energy substrate molecule to ADP with no requirement of the electron movement. Second, acetyl-CoA formation, or pyruvate oxidation. 2 pyruvates from the previous step, glycolysis, at the cytoplasm will be transported into mitochondrial matrix through proton channel. To create acetyl-CoA, each pyruvate is added with coenzyme A tail while removing 1 carbon in forms of carbon dioxide and creating NADH. As a result, 2 pyruvates result in 2 acetyl-CoA with 2 NADH and releases 2 carbon dioxides. Third, Krebs cycle. The 2-carbon acetyl-CoA enters the Krebs cycle by cutting its CoA tail and reacting with 4-carbon oxaloacetate (OAA) to create the first 6-carbon intermediate citrate and isocitrate respectively. Isocitrates remove 1 of its carbon in forms of carbon dioxide to create alpha-ketoglutarate, or alpha-KG, while donating proton to NAD to create 1 NADH. Subsequently, alpha-KG reacts with CoA tails and remove 1 of its carbon in forms of carbon dioxide to create succinyl-CoA, while donating electron and proton to NAD to create 1 NADH. Succinyl-CoA leaves its CoA tails to create succinate. Succinates provide high energy electron to FAD to create fumarate while providing FADH2. Fumarate turns to malate and eventually complete the Krebs cycle by ending with OAA and donates electron and proton to NAD to create 1 NADH. Since cells has 2 acetyl-CoAs, they will undergo 2 Krebs cycles while creating 6 NADH, 2 FADH2, and releases 2 carbon dioxides. Fourth, ETC, electron transport chain. The function of ETC is to convert energy carriers NADH and FADH2 to be ATP. The first step is that NADH donates 3 electrons and 3 protons at the complex 1, leaving with NAD+, and FADH2 donates 2 electrons and 2 protons at the complex 2, leaving with FAD. All protons move to mitochondrial intermembrane space through complex 1 and 2 on the inner membrane. It makes the intermembrane space have high proton concentration. Electron move through Q, complex 3, and cytochrome complex approaching complex 4. With the help of cytochrome complex oxidase, the electron will react with oxygen and proton in the inner membrane of the mitochondria to create water or H2O. Since concentration of proton in the intermembrane space is higher than the inner membrane, it leads to the proton gradient, where proton moves from the higher concentration in the intermembrane space toward the lower concentration in inner membrane through the ATP synthase. ADP with phosphate receive proton to create ATP. In conclusion, 3 protons from NADH create 3 ATP, while 2 protons from FADH2 create 2 ATPs. In total, from glycolysis until ETC, 34 ATP is created through oxidative phosphorylation, and after 2 is created through substrate level phosphorylation during glycolysis. Right now, we understand all process involved energy production. Here, we focus on the effects of cyanides on the cellular respiration. During the ETC, cyanide binds with Fe core or iron core of cytochrome complex oxidase. Therefore, it prevents electron to react with proton to create water. Since water cannot be created, the electron is unnecessary, and NADH and FADH2 do not donate their electrons and protons to the protein complex 1 and 2. The proton concentration in the intermembrane space is lowered to be identical as the concentration of proton in matrix. As a result, it destroys proton gradient, meaning that proton cannot move from the intermembrane space to matrix. Eventually, ATP synthase cannot provide high energy proton to ADP to generate ATP. Even though O2 is present, cell has to use anaerobic respiration to produce ATP, as the oxygen cannot be reduced in matrix due to the binding between cyanide and Fe core. Instead of 36 ATP can be created, only 2 ATP is created through glycolysis in cytoplasm while making lactic acid. Normally, lactic acid leads to the temporary muscle fatigue due to lack of oxygen, so cell can return to aerobic respiration later. However, cyanide binds permanently with the Fe core, which eliminates the chance that cell can return to aerobic respiration, and it has to continue with lactic acid fermentation to produce 2 ATP. Without the treatment, high concentration of lactic acids makes the blood pH lower than 7, or called lactic acidosis. The acidic blood is harmful for human body. To relieve the impact of cyanide exposure, first, immediate actions. Remove the patient from the source of exposure and administer 100% oxygen to support cellular respiration. The high concentration of oxygen can encourage complex 4 to receive oxygen to create water and return cell back to the aerobic respiration. Second, specific antidotes. First, the preferred antidote, hydroxycobalamin. Cyanide ion will bind with cobalt core of cyanocobalamin or vitamin B12 in this antidote, instead of binding with the iron core of cytochrome complex oxidase. So, this reaction will immediately decrease the cyanide concentration in human blood, and the cobalt core binding with cyanide will be excreted through urine. Second, sodium thiosulfate or Na2H2O3. By competing with iron core in cytochrome complex oxidase, thiosulfate will react with cyanide to create thiocyanate HCN-, as shown in the equation. The thiosulfate, which is relatively harmless, is then excreted by the kidneys. Therefore, thiosulfate can decrease the concentration of cyanide and ultimately decrease mortality due to lactic acidosis and cyanide exposure. Thank you.